So to begin our discussion on glucose metabolism, let's take a look at glycolysis. Now, glycolysis is also known as the glycolytic pathway, and glycolysis actually involves many individual steps and many individual processes. And so we typically divide glycolysis into three different stages: stage 1, stage 2, and stage 3. Now in this lecture we're going to focus on stage 1, but before we take a look at the details of stage 1, let's actually discuss what each stage actually does. So what's the point of stage 1, stage 2, and stage 3? Well, stage 1 basically aims to trap that glucose molecule inside the cytoplasm of the cell so that the glucose cannot actually leave that cell. And it also destabilizes that glucose molecule, increases its overall energy, and that makes it more reactive, and that prepares the glucose for stage 2. Now, in stage 2, we break down that destabilized glucose molecule into two components. And then in stage 3, we essentially take those two components, we oxidize those two components, and that allows us to actually harvest the energy in those molecules and form ATP molecules as well as pyruvate molecules and NADH molecules. So let's focus on stage 1. So in stage 1, we basically want to take that glucose molecule and transform it into fructose 1,6-bisphosphate. And we'll see exactly why that is in just a moment. So actually, if we look at stage 1, we can break down stage 1 into three different steps. We have step 1, we have step 2, and we have step 3. So let's quickly go over what each step does and why. So in step 1, we basically phosphorylate one of the carbons on glucose. In step 2, we transform that glucose into an isomer called fructose. And in step 3, we essentially take that fructose and we add another phosphate group onto that fructose. So let's focus on step 1. So what exactly are the details of step 1 and why does it actually take place? So let's suppose we have a glucose on the outside of the cell, and in the membrane of that cell there's a special type of membrane protein that we're going to focus on in a future lecture that actually transports that glucose into the cytoplasm of that cell. Now, once that glucose is inside the cytoplasm of that cell, a special type of enzyme, a protein kinase we call hexokinase, actually catalyzes the addition of a phosphoryl group from an ATP molecule onto the carbon number 6 of this glucose. So this carbon number 6 basically gains a phosphoryl group as a result of the catalyzation of this hexokinase. Now, we'll take a look at the details of hexokinase in just a moment. First, I'd like to answer the following question. What is the entire point of step 1? Why do we have to actually add a phosphoryl group onto the glucose molecule? Well, two important reasons. Number 1 is we want to trap that glucose in the cytoplasm of that cell. So by adding that phosphoryl group onto carbon number 6, we change the structure of this glucose molecule. We transform it into glucose 6-phosphate. So what we do is we basically make it much more polar because now it contains a full negative charge. In fact, a negative charge of 2, whereas in this case, it wasn't as polar because it didn't contain a full charge. And so what that means is those membrane transport proteins that were able to actually move the glucose into the cell or out of that cell cannot bind the glucose 6-phosphate because of this charged component. And so these glucose 6 molecules cannot leave the cytoplasm, cannot exit the cell, and that means they are trapped inside the cell. Now, the second reason for step 1 is to basically begin destabilizing that glucose molecule. So remember, whenever we have charge, that destabilizes the structure. And in this particular case, we have no charge. In this case, we have a charge of -2. So that means this molecule is higher in energy, less stable, and more reactive than this glucose. And that's important in basically destabilizing that structure so that in step 2, it is able to actually react and break down into smaller components. Remember, the entire point of stage 1 is to trap the molecule and make it less stable. So once again, glucose moves into the cell with the help of a membrane transporter. Once inside the cytoplasm, it undergoes a phosphorylation process in which hexokinase adds a phosphate group, phosphoryl group, onto the 6th carbon by taking it away from ATP, and we also form the ADP. So actually this step requires energy, requires an ATP molecule. So this step is significant for 2 reasons. Number 1 is it makes the glucose more polar by adding that negative charge, and this traps the glucose inside the cell. So the negatively charged phosphate group prevents the glucose from moving across the cell membrane because that membrane transport protein cannot actually attach to the glucose 6-phosphate because of this altered structure. Now, the second reason is to begin destabilizing this molecule. That increases its energy and makes it more reactive. So the addition of a charged moiety onto the glucose destabilizes the structure of that glucose and increases its energy. And this makes it more reactive and more likely to actually undergo and continue glycolysis. Now, let's focus in on the hexokinase. So what is a hexokinase? Well, a hexokinase is a protein kinase that basically catalyzes the addition of phosphoryl groups onto hexo sugars, and glucose is an example of a hexo sugar. It contains these 6 carbon atoms. Now, like all protein kinases, hexokinase requires a divalent metal atom to actually function effectively, and that's because the divalent metal atoms, such as magnesium 2+ or manganese 2+, interacts with the ATP molecule. So when the divalent ion interacts with that ATP molecule, it changes the conformation of that ATP molecules and makes it perfect to undergo this reaction in the active site of the hexokinase. So this is hexokinase before the binding of the glucose. This is our glucose, and this is hexokinase after that glucose has bound to the active site. So before the binding actually took place, we see that we have 2 domains, and these 2 domains are found relatively far apart. But when that glucose moves into this pocket, the active site of the hexokinase, these 2 lobes, these 2 domains basically rotate about 12 degrees inward. And as they rotate, they essentially create this induced fit for the glucose. And that means the glucose will fit snugly inside this region. So as it essentially closes in, it basically squeezes all the water molecules out of this region. And so once we form this structure, we have this induced fit, this perfect fit, and all the water molecules have been removed from the active side. So it creates a perfect environment for this reaction to actually take place. So on top of actually creating this close proximity, it also creates an environment in which we don't have any water molecules. Now, why is that important? Well, whenever we have an ATP molecule and we have a water molecule in close proximity, remember the ATP is not very stable, and so the water molecule will generally want to hydrolyze and break down that ATP into ADP. And so by creating this environment in which we don't have any water molecules, what that does is it prevents any unwanted reactions. That is, it prevents the water from hydrolyzing the ATP into ADP. And what that means is the ATP can go ahead and transfer, or the enzyme can go ahead and transfer that phosphoryl group from the ATP onto this glucose molecule, in turn producing that ADP. So the movement of the glucose into the active site of hexokinase causes the 2 domains, these 2 domains, to basically rotate about 12 degrees. So they actually move about 10 angstroms, and that rotates these sections and closes that glucose molecule inside this active site, creating an induced fit. and this seals off the glucose, so removes all the water molecules and positions the 6th carbon right next to that ATP. And that also prevents the ATP from actually being hydrolyzed prematurely by that water molecule. Because if the ATP is hydrolyzed by water before that phosphoryl group is actually transferred onto the glucose, then we form the ADP, and the ADP will not be able to transfer that phosphoryl group. And so that's why this is a very important step. Now, let's move on to step number 2. We call the isomerization step. So once we actually form that glucose 6-phosphate, so glucose that contains a phosphoryl group on carbon number 6, the next step is to actually transform that glucose into a fructose. And to transform it into a fructose, what must happen first is it actually has to open up into its open chain conformation. Why? Well, remember in our discussion of glucose molecules and sugar molecules, we said that when the sugar molecules exist in their cyclic form, they do not typically undergo any reactions because that aldehyde group or the ketone group is not actually exposed. And so what happens is a special enzyme known as phosphoglucose isomerase opens up this structure to form the open chain conformation now that exposes this reactive aldehyde group. In the next step, we basically catalyze the transformation of this glucose in its open chain into a fructose in the open chain. And next, we transform the open chain into its closed cyclic chain. And so this is the final product in step number 2. This is what we call fructose 6-phosphate. So in the second step, the enzyme phosphoglucose isomerase transforms the glucose, which is an aldose, it contains an aldehyde group, into a fructose, which is a ketose that contains this ketone group. So this is a ketone and this is an aldehyde. So we see that in step 2 of stage 1, the entire point is to transform an aldose into a ketose. Now, let's move on to step 3 of stage 1. So it's basically not enough to add that phosphoryl group. We have to add a second phosphoryl group, because remember, the entire point of stage 1 is to destabilize that glucose as much as we can. And so to further destabilize the structure of glucose, to increase its energy and make it much more reactive, in step 3 of stage 1, we have an enzyme known as phosphofructokinase, PFK, that catalyzes the addition of yet another phosphoryl group onto a carbon of that fructose. And so we begin with fructose 6-phosphate, and then we form fructose 1,6-bisphosphate. And so now instead of having 1, we actually have 2 of these phosphate groups. And so just like this step actually requires ATP, this step also requires ATP. And so we see that stage 1 actually uses up 2 ATP molecules, and no ATP molecules have been actually formed so far. Now, another important thing about this step 3 of stage 1, is once the step actually takes place, that commits that sugar molecule to undergoing the glycolysis process, because before this step actually took place, that sugar molecule can be stored in the form we call glycogen. But once this step takes place, we call it the committed step, because it commits that sugar to undergo that process of glycolysis. So in step 3 of stage 1, phosphofructokinase, PFK, adds a second phosphoryl group onto that sugar to make it much less stable, higher in energy, and therefore much more reactive. And that will allow it to actually break down in stage 2 into smaller components, as we'll see in the next lecture. And so we transform the fructose 6-phosphate into fructose 1,6-bisphosphate. And this is what we call the commitment step. It commits it to the process of glycolysis. So Once again, the point of stage 1 is to trap that molecule inside the cell, and that's exactly what we do in step 1. So we add the phosphoryl group to basically trap it inside the cytoplasm. And by adding that phosphoryl group, we also destabilize that molecule. Now in step 2, we transform it into from an aldose into a ketose, and then in step 3, we continue the process of destabilization by basically adding a second phosphoryl group onto that sugar molecule.